All right, so double checking. I'm good. Thank you so much for asking. I'm just checking if you can see both the PowerPoint and the Blackboard on here. And do you hear me clearly? All right, great. All right, so as we have been discussing, we did discuss last class the different parts of the central nervous system. And if you remember, the two main compartments of the central nervous system were the brain and spinal cord. And if you remember, the brain was formed of four main parts. Those were my cerebrum, my diencephalon, my brainstem, and the cerebellum. Last class, if you remember, we did discuss the cerebrum and the diencephalon. If you remember, the cerebrum was formed of two cerebral hemispheres. Each of them has those ridges on their outer surface. Those elevated ridges, if you remember, we called single one of them is the gyrus, and multiple ones are by gyri. The cerebrum was divided into two cerebral hemispheres. And if you remember, between those elevated ridges, we have those depressions. Shallow depressions are what we call the sulcus or sulci. If you remember, we did divide the brain or the cerebrum into lobes. We've got the frontal lobe, parietal lobe, occipital lobe, and temporal lobe. And the insula was located deeper on here at this point because it has a depth to the brain. So again, what are the different lobes? We have 5 lobes of the cerebrum that will be corresponding to the different cranial bones. Remember, we've got frontal, parietal, occipital, and temporal. And here will be the insular lobe or the insula. Those are the different lobes of the cerebrum. If you remember, cerebrum was divided into a gray matter and outer gray matter. Here we're looking at the inside between the two cerebral hemispheres. We'll see an outer gray matter. All this is the outer gray matter. We called it the cerebral cortex. And if you remember, cerebral cortex is going to be responsible to carry on the conscious activities, sensory perception, sensory detection, initiation of the motor movement, memory, problem, and task management. All those going to be taking place in my cerebral cortex. Remember, we did have a set of bundles of myelinated nerve fibers that will be connecting the two cerebral hemispheres. If you remember, each cerebral hemisphere is going to be controlling the contralateral side of the body. So right cerebral hemisphere is going to be controlling the left side of the body. Left cerebral hemisphere is controlling the right side of the body. We called those bundles of myelinated nerve fibers that will be connecting the right and left cerebral hemispheres together. This is the corpus callosum. Located deeper to the corpus callosum, we've seen the other part of the brain in here. This oval shaped structure is what we've called the thalamus. Above the thalamus, we've seen the epithalamus, which is divided into two main parts, the choroid plexus and the pineal gland, if you remember. Below the thalamus, we've seen the hypothalamus that will be connected to a gland pending down from the base of the brain. This is called the pituitary gland. If you remember, functionally, the cerebrum did have different brain areas, like the prefrontal cortex, like Broca's area, which is the motor area of the cerebrum that controls speech. If you remember, also we've seen the precentral gyrus. This is where you're gonna have, and here's this part. And if we're looking up here, it's gonna be all this part. This is where I will have a very important brain area that will be responsible to initiate the voluntary motor movements. This is what we've called the primary motor cortex. Other brain areas that we've seen, other than the motor areas located in the frontal lobe. Here, this is my prefrontal cortex here again. We've got the sensory areas and their association areas located in, distributed in the parietal, occipital, temporal lobes. If you remember, just behind the central sulcus, we've got the primary somatosensory cortex. And just next to it, we've got the brain area that acts as an association area for you to be able to perceive those sensations that you are getting from your skin, your tendons, your muscles. Those are the somatic sensations. We've seen another sensory area in the occipital lobe, if you remember. This was my visual cortex. And surrounding the visual cortex, we've got the visual association cortex. If you remember, what are the sensory areas responsible for, like the primary somatosensory cortex, like the visual cortex, like gustatory cortex, what are those brain areas responsible for? They are responsible to make you aware of the presence of a stimulus. So I can see using my primary visual cortex. But in order to understand what I see, what gives me a perception of the sensation are the association areas. I can feel the temperature by the primary somatosensory cortex. But in order for me to decode those signals to make a meaning, to create a meaning for the sensation I'm receiving, I need to have an association cortex that will be related to the sensory cortex. We'll see in another brain area. Remember, a large brain area located here. This is the Wernicke's area. And remember, this was the area responsible for the language production. What was the difference between Wernicke's and Broca's areas? This is the one that creates the idea here, Wernicke's. This is the ones that will allow you to put the words together in your mind. But for you to be able to speak those words out, you're gonna need to contract a large group of muscles, and who will coordinate those muscle contractions, who will be responsible to determine the amount of air that you're gonna be breathing out, who's gonna be modifying the sounds emitted from your vocal cords. All this is gonna be taking place under the regulation of Broca's area. Remember, the cerebrum was given a nickname. We did give the cerebrum the nickname is, you remember from last meeting? What was the nicknames that were given to the cerebrum? Cerebrum itself. The ones that will initiate the motor movement, the ones that will be responsible to detect sensations, that will be giving meaning to those sensations, the ones that will give you the ideas, the ones that will be allowing you to speak, responsible for awareness. Yes, the president here. And if you remember, the president won't be capable to take care of every single thing in your body. So we've got the thalamus, which is part of the diencephalon. And if you remember, the thalamus here is gonna be determining what should be delivered to your cerebral cortex and what should remain in your subconscious. It's gonna be responsible to take care of signals that are on their way to your conscious mind, to your cerebrum. Remember, the thalamus here was acting as the vice president. Who would be taking care of the basic activities like regulation of the food intake, regulation of your body temperature, regulation of your heart rate, regulation of all the autonomic activities, even participates in creating your emotions. I am responsible to create the emotional behavior. All those, do we need to exert any conscious effort to take care of those? Would even the thalamus be responsible for taking care of this. Those are all basic activities that will allow you to maintain life. Those basic activities are taken care of by the hypothalamus. Today we are starting our new topic, which is the brainstem. Brainstem has three main regions. Those are the midbrain that you see on here, and this bulging part is going to be the pons. Then I have a straight regions that moves down like this, that will be connecting the brain to the spinal cord. This is my medulla oblongata. So we've got three main regions in the brainstem. Those are midbrain, pons, and medulla oblongata. First one is going to be the midbrain. It's going to be located between the diencephalon and the pons. This is where I have the centers responsible for eye and head movement, the reflex center for eye and head movement. So if you are looking at your screen right now and you decided to rotate your head to the right, would you lose sight of the screen? If you are looking at the screen and you decided to rotate your head to the right side, would you lose the sight of the screen? What do you think? Hello? Anybody? Yes, you will lose if you kept looking at the screen and you are rotating your head. Would you lose sight? What do you think? No. Why? Because what happened in here? Your eyes did rotate at the same speed, but in the opposite direction. My head is rotating, my eyes are rotating in the opposite direction at the same speed for me to keep the same picture that I'm looking at. So did you decide to rotate your eyes in the opposite direction to the head for you to not lose sight of what you're looking at while you are rotating your head? Did you decide to do so? Or is this just a reflex? What brain part is going to be responsible to carry on those head and eye reflexes? So this is going to be your midbrain. Also, we're going to have the cerebral aqueduct that will be traveling, connecting the third ventricle to the fourth ventricle. If you remember, what are the ventricles? The ventricles, those are the spaces filled with cerebrospinal fluid. We did have two embedded in the cerebrum. Those are my lateral ventricles. And if you remember, last class when we discussed the diencephalon, we've mentioned that those are going to be draining down into a third ventricle located in the diencephalon. This third ventricle is going to be connected to another ventricle, another space where the cerebrospinal fluid is going to be traveling, which is going to be located between the pons on one side. If this is a pons like this, and medulla oblongata, and the cerebellum on the other side. So located between your pons and medulla oblongata on one side and the cerebellum on the other side, you've got space filled with cerebrospinal fluid. What do we call this space? This is my fourth ventricle. How about the third ventricle? The third ventricle, if you remember, this was embedded in the diencephalon. So the third ventricle is connected to the fourth ventricle by a duct. You see this duct on here? This duct is traveling in which part of the brain that will be located above the pons? What do we call this part that connects the diencephalon on one side to the pons on the other side? Midbrain, exactly. So we call this duct that will be connecting the third ventricle to the fourth ventricle. This is my cerebral aqueduct. Aqua is water. So cerebral aqueduct is the duct that will be allowing the fluid, cerebrospinal fluid, to travel down from the third ventricle embedded in the diencephalon and the fourth ventricle located between pons medulla on one side and the cerebellum on the other side. All right, this is how the whole brain can look like. And we're looking on the inside of the brain. Moving down to the pons is going to be forming part of the anterior wall of the fourth ventricle, as you see on here. This is my fourth ventricle. The anterior wall of the fourth ventricle is partly formed by the pons. Pons is going to be the main, it's going to be mainly a conducting area between the medulla and the spinal cord below it, and the midbrain diencephalon and the cerebrum above it. Also coming out from the pons, we've got the nuclei for 3 of the cranial nerves. Those are cranial nerves 5, 6, and 7. Those are my trigeminal, abducens, and facial nerves. The medulla oblongata is going to be also sharing in the formation of the ventral wall here, the anterior wall of the fourth ventricle. It's going to be transmitting the ascending and descending impulses back and forth from the brain to the spinal cord and vice versa. In the medulla oblongata, we're going to have the nuclei responsible here for regulation of the respiratory rhythm, regulation of the heart rate, blood pressure. Also, we've got the centers for coughing, sneezing, swallowing, and vomiting. Moving on to our last part of the brain today, this is going to be the cerebellum. And the cerebellum is going to be representing a large part of the brain, but remember, the largest part of the brain is going to be the cerebrum here, which represents around 40% of the brain mass. Here, the cerebellum is going to be representing only 11%. It's going to be located, as you see on here, dorsal to the pons and the medulla oblongata. The main function of the cerebellum is to subconsciously provide precise timing and appropriate patterns for the skeletal muscle contraction. This is what's going to be allowing you to maintain a balanced movement. If you have no idea what is the force of contractions that you're gonna need to generate in order for you to walk, there will be no coordination between your skeletal muscle contractions, and so you're gonna lose the ability to maintain your body balance. Cerebellum is gonna be formed of two cerebellar hemispheres. Don't confuse the cerebellar hemispheres with the cerebral hemispheres. We've got two cerebellar hemispheres that will look like this. And both of them gonna be connected by the vermis. Vermis here is the connection between the right and left cerebellar hemispheres. We're gonna be dividing the cerebellum into 3 lobes, unlike the cerebrum was divided into 5 lobes. Remember? Cerebellum gonna have an anterior lobe, posterior lobe, we've got here anterior lobe, posterior lobe, and the third lobe on here is gonna be my flocculonodular lobe. So 3 lobes that will be present forming my cerebellum. If you see on here, if you get closer to the internal structure of the cerebellum, you will see that it has an outer gray matter and an inner white matter. What does the white matter looks like for you? What does it look like in real life? In your daily life, does this look like? Leaf, yeah, leaves, like a leaf. Branches of a tree, it looks like a tree, you see on here, this is the tree and those are the branches. Can you imagine a tree on here? So what do we call the white matter of the cerebellum? It's the tree of life, arbor vitae, tree of life, arbor vitae. So again, what we're looking at in here, we're looking at the structure and the function of the cerebellum. Cerebellum is formed of right and left cerebellar hemispheres. Both are connected in the middle here by the vermis. We're going to be dividing the cerebellum into 3 lobes: anterior lobe, posterior lobe, and flocculonodular lobe. It has an outer gray matter and an inner white matter, like the cerebrum. Remember, what do we call the gray matter of the cerebrum? What do we call the gray matter of the cerebrum? Anybody? What do we call the gray matter of the cerebrum? Gray matter of the cerebrum. Not cerebellum and not the white matter. We call it the cerebral cortex. And what did we call the white matter of the cerebrum that attaches the 2 cerebral hemispheres to one another? Corpus callosum. Corpus callosum. What did we call here? The white matter of the cerebellum, not the cerebrum, cerebellum. What do we call the white matter of the cerebellum? Tree of life, arbor, right. Easy, easy. Anybody is confused here? Questions, questions? All right, so moving on to the brain ventricles. Again, how many ventricles do we have? How many ventricles do we have? We've got 4 ventricles. Remember, 2 are located in my cerebrum. Those are my lateral ventricles. One is going to be embedded in my diencephalon. We call this is the 3rd ventricle. And one is going to be located between pons medulla on one side and the cerebellum on the other side. This is my 4th ventricle. So again, we've got 4 ventricles. And again, what are those ventricles? Those are spaces filled by cerebrospinal fluid. Cerebrospinal fluid. If you remember, what type of neuroglial cells we've seen lining those spaces that are filled with cerebrospinal fluid? Do you remember what are the different neuroglial cells that we've seen? We've seen a cell that looks like a star. We call those are astrocytes. And what was the function of the astrocytes? Remember, it has arms that will be connecting the blood vessels and other arms that will be connected to the nerve cells. They are acting as a blood-brain barrier. They are helping with the metabolism of the nutrients. We've seen microglial cells, which are much smaller in size, that are defensive cells that will be attacking any bacteria or virus that finds its way towards the nervous system. We call those are microglial cells. We've seen another type of neuroglial cells, and those are the oligodendrocytes. And if you remember, oligodendrocytes, those are the cells that will be responsible for the myelination of the nerve fibers inside the CNS. And we call them oligodendrocytes because they have few processes. We've seen another cell located also in the central nervous system, and those cells that have cilia. And the propulsion of the cilia is what allowed the cerebrospinal fluid to circulate within the ventricles, and we call those are my ependymal cells. So again, what kind of cells do we have lining those ventricles? Those are my ependymal cells. If we're looking on here, this is gonna be my, those gonna be my lateral ventricles. Each of the lateral ventricles has 3 horns. I have anterior horn, posterior horn, and on the side here you've got the inferior horn. So again, each of the 2 lateral ventricles will have 3 horns: anterior, posterior, and inferior. Third ventricle, as you see on here, it's embedded in the diencephalon. Fourth ventricle, as you see on here, is going to be located between the pons medulla on one side and the cerebellum on the other. And if you remember, a duct that will be connecting both third to fourth ventricles passing through the midbrain, we call this is my cerebral aqueduct. So we can see on here 2 C-shaped ventricles. Those are my lateral ventricles. Both are draining down into third ventricle. Third ventricle drains into fourth ventricle. Again, what's connecting this third to the fourth ventricle is going to be a duct traveling through the midbrain, and this is going to be my cerebral aqueduct. Questions? All right, so now we're moving to the functional brain systems. We are looking at, in this chapter, 2 brain systems. First one is going to be the limbic system. Second one is the reticular formation. Limbic system is going to be formed of structures of the brain that are located on the medial aspects of my cerebral hemispheres and the diencephalon. Those will include parts of the diencephalon and some structures that will be encircling the brainstem. The main function of all those brain areas would be to produce emotions like anger, fear, conflict, and according to those emotions that those brain areas are going to be producing, this will result in a modification of the behavior. So if you are afraid, for example, you're going to behave in a different way than if you are relaxed. You have nothing to worry about. So what did cause this modification of the behavior is the emotions. Like what brain areas on here, we don't need to memorize those brain areas that share in the formation of the limbic system, just get a general overview. Parts include cingulate gyrus, the septal nuclei, the amygdala, the hippocampus, the dentate gyrus, parahippocampal gyrus. All those are different brain areas that are responsible to produce emotions. Again, this is a diagram that you don't have to worry about for the upcoming lab exam. All right, this is not an important diagram. At the end of class today, I will go over the most important diagrams that you should be ready with for the last lab exam. Moving on to another brain system is going to be the reticular formation. Reticular formation is formed of three broad columns of nuclei that will be located along the length of the brain stem. So they are sets of nuclei located in the brain stem. They will allow the signals to travel to the hypothalamus, to the thalamus, to the cerebral cortex, to even the cerebellum, coming from mainly the spinal cord. So I'm acting here to distribute the signals to the different brain areas. And the main function here of this brain system is going to be to maintain the alert state of the cerebral cortex. If a severe damage did affect, if a person did have a severe damage to the reticular formation, to the brain stem, where the reticular formation nuclei are going to be kept, this is going to result in permanent unconsciousness, or what we call coma. So if we're looking on here, those are my reticular formation nuclei located in the brain stem. And they are sending out the signals to almost everywhere in the brain to maintain an alert state. Like what signals would they respond to? Like, for example, touch, pain, temperature, light, visual impulses, auditory impulses, sounds. All those types of stimuli will enhance the reticular formation to maintain an alert state of your cerebral cortex. So think of how a person would wake you up in the morning. First, this person will call your name, auditory impulses. So if you didn't respond, you are not waking up. So he will turn on the light. For you to wake up. He will touch you, then will give you a punch in the face for you to wake up. So touch, pain, stimuli also going to be stimulating the reticular formation to activate and maintain an alert state of your cerebral cortex. Also temperature. Have you ever waked up in the middle of the night just to to modify the AC temperature all the time. So why did I wake up? Simply because the temperature sensation here, the time detecting, did activate the reticular formation, those nuclei located in the brainstem. And once they become activated, they will start to send signals to the cerebral cortex, and this will wake me up during, just to modify the AC temperature. All right, looking on here at the protections that we do have for the brain. From outside in, you've got the skin covering the outside of your cranial bones, scalp with the hair. Underneath it, you've got the bone, cranial bones. And underneath the bone, you're gonna have 3 membranes. Those 3 membranes are gonna be your dura mater, arachnoid mater, and the pia mater. 3 membranes, those are together called the meninges. Those are my meningeal membranes. Also remember, the cerebrospinal fluid is not only located inside those ventricles, lateral, third, and fourth ventricles, they are also, the cerebrospinal fluid is also going to be circulating around the brain. So I have a watery cushion formed by the cerebrospinal fluid around my brain. Also, if you remember, we've got some degree of protection from anything that travels from the blood to the nerve cells, having those astrocytes located in between the blood vessels and nerve cells, acting as blood-brain barrier. Moving on to the meninges on here, they contain the cerebrospinal fluid. We have 3 layers from outside in. We've got the dura mater, arachnoid mater, and the pia mater. Looking on here, this is going to be the skin of the scalp. Then we've got the bone, cranial bone. And remember, around any bone, you've got the periosteum. You've got periosteum on both sides of the cranial bone. Underneath it, and actually as part of my periosteum, part of the periosteum acts as a layer of the dura mater. This is the first meningeal membrane. Underneath the dura mater formed of the internal periosteum and the meningeal part, all this is my dura mater. Underneath it, you've got a web, spider web shaped layer containing spaces that has blood vessels in. This is called my arachnoid mater. And we've got a very delicate membrane that will be covering the outer surface of the brain. This is my pia mater. pia matter. So again, what are the layers from outside in? You've got first scalp, then you've got the periosteum. Below the periosteum, you've got the bone of the skull, the cranial bone. Then you've got the dura mater, arachnoid mater, and adhering to the outer surface of the brain is going to be the pia mater. Cerebrospinal fluid is a watery solution that will be circulating inside the brain ventricles and around around the brain and spinal cord. If we look on here, this part of the brain is what we've called the thalamus. And remember, above the thalamus, we've got the epithalamus. If you remember, two parts that were forming the epithalamus. We've got the pineal gland, which was responsible for the release of the melatonin that regulates the sleep-wake cycles. And we've got the choroid plexus. And if you remember from last class, what was the function of this choroid plexus? It was to produce the CSF, to produce the cerebrospinal fluid. So this cerebrospinal fluid is going to be circulating in the lateral ventricles, then gets drained to the third ventricle located in the diencephalon, then travels through the cerebral aqueduct to reach my fourth ventricle, which is going to be located between the pons medulla on one side and the cerebellum on the other side. Part of the fluid here leaves the brain and will be circulating around the brain in the subarachnoid space. And another part will be leaving to circulate around my spinal cord. Part of the cerebrospinal fluid also travels in the central canal of the spinal cord. So again, where do I produce the cerebrospinal fluid? The cerebrospinal fluid is going to be produced from your choroid plexus, which is part of my epithalamus. It circulates inside the ventricles, lateral, third, and fourth ventricles. It also circulates in the subarachnoid space. This is where the cerebrospinal fluid gonna be drained through those villi. We call those are the subarachnoid villi. This fluid gets drained to your venous circulation. All right, so which one do you think should be greater? The rate of production or the rate of drainage of the cerebrospinal fluid? They should be equal. Why they should be equal? To maintain a constant volume. Try to imagine that the rate of production is greater than the rate of drainage. What's going to happen? The ventricles in the brain going to be dilated, and this is going to compress the brain tissue. All right, for the in-class activity, first question for today is going to be to put those protective layers in order from outside moving inwards. So which one is going to be located outside, closer to the bone? Dura mater. Dura, remember, strong, tough. What comes next is going to be the one that has spaces and contains the blood vessels. This is my arachnoid mater. So you've got 2, 3, and the delicate membranes that's going to be adhering to the outer brain tissue. This is going to be my pia mater. So the good answer here is going to be D. All right. Any questions? Any questions? Let's move on to the structure of my spinal cord. Spinal cord is going to be formed by spinal nerves. We have 31 pairs of spinal nerves. If we're looking here at the spinal cord, you will see spinal cord has 2 enlargements, an enlargement in the cervical region and another enlargement in the lumbar region. Why do we have those enlargements? Because I need a lot of nervous tissue where I get the nerves coming out to reach my limbs. So the nerves here are coming out. You've got more nerves coming out from the cervical region, from the lumbar region, for you to innervate both the upper and lower limbs. Towards the end of the spinal cord, you've got something that looks like a tail of a horse, which are nerve roots located inferior at the inferior end of the vertebral canal. So you see here, this is the tail of the horse that we're talking about. It's formed of just nerves, just nerves. You don't have spinal cord below this point. All right, so what do we call this tail of the horse? We call it the tail of the horse, coda equina. Coda equina is tail of the horse. Spinal cord will have tracts that will act as pathways. We've got pathways traveling up to the brain. We call them the ascending pathways. And we've got signals traveling down through tracts. So those are my descending pathways, which are going to innervate my muscles and glands. Spinal cord protection is going to be very similar to the ones that we've seen with the brain. So to the outside, you've got the bone. We don't have cranial bones here, but we do have the vertebrae. We've got the 3 meninges, which will keep going surrounding the spinal cord. And we've got this cerebral spinal fluid as well. That will be still located in the subarachnoid space below in the arachnoid matter. An extra protective layer that I have here around the spinal cord, but is not located within my around the brain, which is going to be the cushion of fat and network of veins that will be located above the dura mater in the epidural space. So if we're looking on here, what are the protective layers from outside in? If I'm not naming them, first layer outside is going to be my skin, definitely. Next is going to be my bone, which is going to be the vertebrae. To the inside of the bone, you've got the meninges, remember? Dura mater, then arachnoid mater, then you have the pia mater. But you've got here in an epidural space, a space above the dura mater, which contains fat and veins. And in the subarachnoid space, same like around the brain, you've got the cerebrospinal fluid, the CSF. So if I'm looking here at the protective layers of the spinal cord from outside in, you've got the bone, you've got the fat and veins in the epidural space, you've got the dura mater, then arachnoid mater, then you've got this watery substance that will be circulating around the spinal cord, CSF. And then you've got your pia mater adhering to the outer surface of the nervous tissue. If we're looking on here at the structure of the spinal nerve and its location, if you see on here, this is going to be my spinal cord. It has an H-shaped gray matter, and coming out of the spinal cord, you've got the spinal nerves. Each spinal nerve, if you notice, has a dorsal root and a ventral root. Both will fuse together to form the spinal nerve. So again, dorsal root, ventral root, forming together the spinal nerve. Looking closer to the structure of those H-shaped gray matter. Each H-shaped gray matter are gonna have a ventral horn. You see on here, this is my ventral horn. Also, it's gonna have a dorsal horn. And only in the thoracic region, you've got the lateral horn. So again, throughout the spinal cord, you've got a dorsal horn, a ventral horn, and only in the thoracic region, you've got the lateral horn. Is there any difference here between the three horns in terms of function? Can you remind me, what did we mention about the gray matter? What is forming the gray matter in your central nervous system? Cell bodies and unmyelinated nerve fibers. Exactly. So we're looking here, those are cell bodies. They are either gonna be sending up towards your brain signals, so a sending pathway, or they are sending out signals to muscles and glands. So descending pathway. So the nerve cells that will be sending out the motor impulses to reach the target organs here, those are going to be located within your ventral horns. the ventral horns of the gray matter of the spinal cord. So this is where I've got the nerve cells dedicated to send out the somatic motor signals. The voluntary motor movement takes place by conducting the signals to my skeletal muscles from the spinal cord. And again, to be more specific, what part of the spinal cord gonna have the nerve cells responsible for the conduction of this kind of signals is gonna be the ventral horn of the gray matter of the spinal cord. What if I'm receiving signals from receptors? You might receive signals from your stomach, right? We call those are visceral sensations. You might receive signals like pain, temperature, pressure from your skin, from your muscles. We call those sensations, those are somatic sensations. So the receptors detecting different sensations here will be conducting their signals towards, this time, not the ventral root of the gray matter. They will be conducting their signals towards the dorsal root of the gray matter. For the somatic sensations, those will be conducted to the end, to the posterior part of the gray matter of the dorsal root of the gray matter of the spinal cord on here. Compared to the visceral sensations coming, for example, from blood vessels, from stomach, from the lung tissues, they will be conducted towards the nerve cells located in the anterior part of the dorsal root of the gray matter of the spinal cord. How about my motor signals I'm sending down from the brain to reach my smooth muscles, cardiac muscles, or my glands? I don't have any control. They are not somatic motors. They are autonomic. So the nerve cells that are going to be conducting autonomic motor signals, they are going to be kept in your lateral horns of the gray matter. And again, lateral horns are going to be located in your thoracic region of the spinal cord. Questions? Questions? All right, so looking at the outside part on here, I am full of white matter. The white matter is formed by myelinated nerve fibers. And those myelinated nerve fibers are going to either be conducting signals up towards my brain, or they are carrying signals down from the brain to reach the target organs. So here, if I'm conducting signals up and down, those signals are going to be traveling through my white matter. If the signal is going to be getting in or out from the spinal cord, it's going to be getting in and out from the gray matter. This is where I have my nerve cells, the cell bodies of the nerve cells. Questions? Questions? Looking at some homeostatic imbalances. First, we're looking at the loss of function of the nervous system. If I have sensory loss, if I lost the ability to detect sensations, this is called a paresthesia. If I lost the motor function here, it's called a paralysis. So again, lost sensation, it's gonna be paresthesia. If I've lost the motor activity, it's gonna be a paralysis. We have two types of paralysis. First, we need, in order to understand the difference between the two types, we need first to understand where and how the signal gonna be conducted to reach my skeletal muscle. So remember, what was the brain area responsible to initiate the voluntary motor movements? Yeah, it was located in the frontal lobe, but what exact area in the brain? The primary motor cortex, exactly. So I've got nerve cells, cell bodies, that are going to be initiating the voluntary motor movement in my primary motor cortex. And those nerve cells going to be traveling down. Remember, it crosses to the other side to reach the spinal cord. And here we're looking at a cut section of the spinal cord. This is how the spinal cord is going to look like. This nerve cell is traveling first through tracts. Remember, if you're conducting the signal up and down, it's going to be through myelinated nerve fibers. It's going to be through tracts in the white matter of the spinal cord. Until where? Until I reach the level in the spinal cord that I need to exit to reach my target organ. So for example, if I'm sending out the signal to my biceps brachii, the signal will exit in the cervical region. So I need to conduct the signal to another nerve cell that will be reaching the biceps brachii. Where this nerve cell is going to be located, it's located in the ventral horn of the gray matter of the spinal cord. What do we call this first nerve cells that travels all the way from the primary motor cortex to my spinal cord? This is my upper motor neuron, compared to the one that exits the spinal cord coming from the ventral horn of the gray matter. This is my lower motor neuron. Is this the only kind of signals that will be reaching this biceps? Actually not. If you tap by a hammer on the biceps, stretching the biceps, and once the biceps got stretched, receptors within the biceps will send out signals to the spinal cord that causes the innervation of the lower motor neuron, causing the muscle to contract. So it's a reflex. You did not decide to contract the muscle. So this upper motor neuron, if a person got a stroke, I did block the blood supply going to the cerebral cortex, so the nerve cells that gets affected is going to be my upper motor neuron. In an upper motor neuron lesion, I'm not only going to be losing the excitatory signals coming down from the brain, I will also lose the inhibition coming down from the brain. So who will take the upper hand is this reflex excitation. This results in a continuous contraction of my skeletal muscle. We call this is a spastic paralysis. Can I decide to contract this biceps? Can I perform a voluntary motor movement? No. So it's a paralysis. Is the muscle contracted or relaxed, fully relaxed? If it's contracted, it's a spastic paralysis. If you're looking on here to this kid, you will see hyper contraction of his muscles. He has what kind of paralysis here? He has spastic paralysis. So this is not a stroke. This is called cerebral palsy. Cerebral palsy, it's most likely going to be due to difficulty during labor. And as a result of this difficulty, the blood supply going to the brain has been disturbed. This disturbed blood supply to the brain going to result in loss of those important cells, the upper motor neuron cells. And if I don't have upper motor neurons, I cannot decide to contract the muscle, and I will have hyperexcitation of the muscle, which will cause a exaggerated reflexes, and hypercontraction. Is this a voluntary contraction? Is this kid is willingly performing those muscle contractions? No. What if a person got an spinal cord injury, car accident that caused a cut in his lower motor neuron? Can I perform a voluntary contraction of the muscle? Would I be able to send out a signal coming down from the brain to the spinal cord and then exits the spinal cord to reach the muscle? No. So this is going to be a paralysis. How about the reflex? Is the reflex is intact? Can I, when I stretch this muscle, respond to this stretch, or is it gonna also be disturbed? See, you've cut the lower motor neuron. So even the reflex won't work. So I have a complete relaxation of the affected muscles. We call this is a flaccid paralysis. So in a flaccid paralysis, what will I see? I see no impulses reaching my skeletal muscles. Do I have any control? No voluntary or involuntary control of the muscles. Neither reflexes or voluntary control. So what happens over time, if those muscles are not performing any kind of contraction, they will atrophy. They will pass through atrophy. How about spastic paralysis? Spastic paralysis is going to be due to damage of the upper motor neuron. An upper motor neuron lesion gives the reflexes the upper hand. I'm not going to be inhibiting any of the excitatory impulses resulting from the reflex arc. This damage gonna cause the muscles to perform exaggerated contraction. Do I have any voluntary control of the muscle contraction? No voluntary control. It's a paralysis. Moving on to spinal cord traumas. So in a spinal cord trauma, if a person got a transection of the spinal cord, so he loses all those roots, ventral, dorsal roots at this level of the transection. If the transection is in the cervical region, this is going to result in quadriplegia, paralysis and loss of sensation of all four limbs, quadri. What if the transection took place between T1 and L1, between first thoracic and first lumbar? So if the transection takes place in this region, this is going to cause the loss of sensation and motor function, voluntary motor action of both lower limbs. So we call this is my paraplegia. Questions? Questions? All right, so we're going to be skipping over the homeostatic imbalances here. Please read this on your own, and if you've got problems with any of them, please let me know next meeting. So after break today, we're going to start our discussion of the eye and ear, which are very important for our upcoming lab exam. Again, the last lab exam is going to be available for you to complete between this Tuesday and the following Monday. All right, so again, this is going to be our last lab exam for this semester. When we come back from the break, we're going to be covering a very important part that will be covered on this lab exam. Any questions for me? Any questions for me before we move on to the break? It's going to be available starting this Tuesday. On Tuesday. It's going to be the same exact structure like the muscles lab exam. It's going to be the same exact structure like the muscles exam. No, it will not be proctored. It won't be proctored. Any questions before we move on to the break? All right, so we can have a break for 10. We come back in 10 to start our discussion of the special senses. No, we're not carrying over any topics to the next semester. So we're still gonna have 2 more meetings before the end of the semester. We're still gonna be completing some of the important topics: endocrine system, autonomic, and peripheral nervous. Those are gonna be completed in our next 3 meetings. No, nothing gonna be carried to the next semester. All right, welcome back. Today we are starting our discussion of the special senses. We will see here the different parts of the eye. We're going to have an understanding of the physiology of the vision, how vision takes place. We're going to understand how the sense of smell is produced, and we're going to learn the structures and functions of the ear, and we're going to discuss together the physiology of hearing and equilibrium. Starting first with the vision. First, before we start our discussion of the vision, we need to have a general overview of the structure of the eye. If we're looking on here, you will see that the eye is going to be kept inside a bony socket. This is my orbit. And if we see on here, we're going to have muscles that will be attached to the eye lobe, to the eyeball. And you will see that the superior and inferior eyelids are covering the anterior surface of the eye. If we're looking on here, the inside of the eyelid, either the superior or inferior eyelids, along with the external part of the eyeball, they will be covered by a membrane. We call the membrane here is the conjunctiva. We call the part of the conjunctiva that covers the eyeball, this is the bulbar conjunctiva. And the part of the conjunctiva that will be lining the eyelid from inside, this is my palpebral conjunctiva. If a person got an inflammation of the membrane here, that will be lining the eyelid and covering the eyeball from outside, we call this is a conjunctivitis. If we see the two parts of the conjunctiva, we'll see that we have a part of it that is neither covering the eyeball from outside or lining the eyelid from inside. We call this is my conjunctival sac. What is the main function of the conjunctiva here? It's going to be to release the mucus secretions that will be lubricating the eye from outside and reduce the friction between the eyelid and the eyeball, preventing the formation of ulceration to the cornea, for example. Looking at the structure of the eyeball, we will see that the eyeball itself is going to be formed of 3 layers. An outer fibrous layer, we call this outer fibrous layer that will be opaque, forming the main skeleton of the eyeball. This is the sclera. And the anterior part of this fibrous layer is going to be clear. We call this is my cornea. Deeper to the fibrous layer formed by the sclera and the cornea, we've got the vascular layer. This is the layer that contains the blood vessels that will give the nutrition to the sclera and the cornea, as well as to the internal layer, as you're going to see in a second. The vascular layer has an anterior part. This is called the iris. And the iris, as you see on here, has a hole through which the light ray is going to be traveling. We call this hole located in the iris is the pupil. Surrounding the iris, we've got the ciliary body, that will be holding the lens of the eye in its place by very thin ligaments. We call those are the suspensory ligaments. And again, what do we call this part that surrounds the iris? It's my ciliary body. And this clear part through which the light is going to be traveling and converge to fall focused on the retina. This is the lens of the eye. As a continuation of the vascular layer lining the remaining part of the sclera, we call this internal lining of the sclera. This is my choroid. What choroid did you hear about just before the break? What choroid did you hear about? Did you hear the word choroid before? Choroid plexus. What was the choroid plexus? Do you remember what was the choroid plexus? It was part of, remember, the diencephalon. To be more specific, it was part of my epithalamus of the diencephalon. And if you remember, the choroid plexus was the part of the brain that's going to be producing the cerebrospinal fluid, the CSF. Here, it's not the choroid plexus, it's the choroid, just the choroid. This is the part, the vascular part on here that's going to be lining the inside of the sclera. Deeper to the choroid, you've got the neural layer. Which mainly going to be formed of the retina. The retina on here, this is going to be the nervous layer that will be responsible to detect the light and transform this light into electrical impulses that will be conducted to the brain through cranial nerve number 2, which is my optic nerve. Again, the inside on here, it's my retina. We divide the inside of the eyeball into two main segments. From the cornea to the lens. We call it the anterior segment. From the lens until you get to the retina, we call this segment of the eye, it's the posterior segment. The anterior segment is going to be filled with a watery substance. We call this watery substance filling the anterior segment of the eye. This is my aqueous humor. Compared to the posterior segment of the eye is going to be filled by a gelatinous substance that will be like a glass. We call this is my vitreous humor. If you're looking right now at the screen of your device, please focus on this point on here. Just focus on this point. Look at this point, this X inside the circle. Can you still see this bird? If you look at this X, can you still see this bird? What do you think? Yes or no? Is it with the same quality? Not in details. Is it with the same quality? What do you think? If you're looking at this X here, you still see the bird, but not with the same quality, right? Not in detail. Why? Because what you are looking at gonna be focused on part of your retina that has better vision. We call this is here. Not sure what's going on. We call this region on the retina the macula lutea. Macula lutea. And it has a central part in it. This is called the fovea centralis. Fovea centralis is this central part of the macula lutea. So in order for you to get an idea about what you are able to see on here, if this is the eye, like this, I have a superior view of the eye and the vision field. The vision field is emitting light that travels like this in the form of light rays. All of the light rays will fall in on different parts of your retina, like what you see on here. So the retina here is gonna be collecting the light from the different parts of the vision field. This is everything that I can see. This is all the vision field. Each part of the vision field is gonna be emitting light, and those light rays gonna fall on different parts on my retina. So the part that you are looking at in the vision field, this axon here, if you decided to look at this X, this means that the light rays are falling on the part of the retina that can see better than the rest of the retina. What do we call this part of the retina again? This is my macula lutea, and the central part of it that can see much better than the rest of the retina is going to be my fovea centralis. Compared to this bird, the light rays that are coming from this bird are simply falling on my retina, still falling on my retina, but away from the fovea centralis. So can I still see this bird? Yes, you can still see it, but not as good as what you actually looking at. As Olivia said, it's the peripheral vision. All right, so if we're getting closer to the retina, how's the retina gonna look like under the microscope? Again, what we're looking at in here, this is the eyeball from inside. And I'm getting very close to check the different layers of the retina. I will see a layer of pigmented cells. They have pigments. And the function here of those pigmented cells is to absorb the light that was not detected by the nerve cells in the retina. Another layer that I can see just in front of the pigmented cells are going to be the receptors responsible to detect light. And we've got two main types of those receptors. I have receptors that will look like rods and other ones that will look like cones. So I have those receptors that will be responsible to detect light. So we're going to call them photo, means light, receptors. And again, how many types of photoreceptors, main types of photoreceptors do we have? We have ones that look like cones and ones that look like rods. So we've got the rods and cones. So I've got light rays traveling like this. So what happens? Those rods and cones will be able to detect the presence of those light rays, and they will start to create an electrical impulse. This electrical impulse is going to be traveling to another layer of cells, nerve cells. Each of those nerve cells has two processes. One that will be acting as a receptive region, one that acts as a dendrite, and the other one acts as an axon. So those cells that will have two processes coming out, those are what we call the bipolar cells. And this is going to be the bipolar cell layer. So you've got an electrical impulse and action potential here that will be produced from those receptors. According to what? According to the light wavelength they will be receiving. So I've received red, I've received blue, I received green. Those colors, those lights gonna be stimulating the photoreceptors to form electrical impulses. So now the electrical impulse is traveling from my photoreceptors to my bipolar cells. So you see, electricity is moving in the opposite direction to my light. So light is moving in, electrical impulse is moving out. Those bipolar cells gonna be then conducting their signals to another type of nerve cells that will have very, very long axons. And those axons all together coming from everywhere in the retina, they will form together the nerves that will be sending out the signals to your brain. So the axons of those cells receiving the electrical impulse from the bipolar cells will be forming the optic nerve. So if I'm trying to draw the different layers of the retina in here, so you've got pigmented cells, then you've got your photoreceptors, either rods or cones. And remember, those photoreceptors will be conducting the signals to nerve cells that did have two processes. We call those are my bipolar cells. Then the bipolar cells is going to be conducting the signals to nerve cells that did have long axons. And the long axons from those cells are going to be forming together from everywhere from the retina, forming together the optic nerves that will be conducting the electrical signals to the brain. Can you remind me what part of the brain is going to be responsible to detect those signals as something that you can see? What brain area? What's the name of the area, specific area in the cerebral cortex? Anybody still here? Exactly, primary visual cortex. This is the one that will be detecting that you can't see. And if a person got a stroke affecting the primary visual cortex, I will have vision loss, will have blindness. Looking here closer to this part in the retina. This part in the retina doesn't have photoreceptors. It has only the axons of those cells. And we call those cells, by the way, here, those are my ganglion cells. So this part in my retina doesn't have photoreceptors. It's only formed of the axons coming from the ganglion cells. We call this region in the retina, this is my optic disc. So how come you do have an optic disc in here, let's say. So this should be corresponding to a region in your vision field. So you see the light rays coming here from this region should not be detected because simply this part on my retina doesn't have photoreceptors. So no photoreceptors means you cannot transform the light rays into electrical impulses. So how come I don't have a vision field defect like this one, for example? So here is a person who has a vision field defect. He has part in his vision field that can detect light. How come I don't have something similar in here? Although I do have an optic disc, or what we call the blind spot. Why we don't have this defect in the vision field, although we do have this blind spot, the optic disc? What do you think? Any ideas? Any ideas? Why is it not present all the time, Najib? If we do, our brain adapts. How it adapts to it? Fills the gap. How it fills the gap? All the time, your eye is actually moving, right? All the time, your eye is moving. All the time, I have movement in my eye. So what happens to this blind spot? It's gonna be receiving light from this region, then from this region as my eye moves, then from this region, then from this region, then from this region. So what happens? You are not capturing a single picture with your eye. You are recording a video. Think of it this way. You have very, very fast pictures that will be detected. And the blind spot gonna be changing its place with every picture. So as my brain, so this is my, so if this is a picture I'm receiving from the, I'm sending to the brain, so the vision field defect is supposed to be here. Then the next picture I'm sending to the brain, it moved in here, then it moved in here, then it moved in here. So my brain now will be overlapping all those pictures together and will be filling the gaps exactly as you've mentioned. Because I know what's in here from those pictures. I know what's in here from those pictures. So my brain will, by overlapping all those images together, it receives from the eye, it will be able to fill up all those gaps in the perceived or the received pictures. Does this make sense? Make sense? The main thing that allows you, that prevents you from having this vision field defect due to the presence of the blind spot is the continuous movement of the eye. Looking on here, what's going to be the direction of the light passing through the retina? First layer that it passes through is going to be the ganglion cell layer. Next is going to be my bipolar cell layer, and then we've got the photoreceptors, and finally we've got here the pigmented cell layer. Compared to the electrical impulse, what's going to be the direction of the electrical impulse? It travels from the photoreceptors to the bipolar cells, then to the ganglion cells. It moves in the opposite direction. All right. Easy, easy. All right, so moving on to the difference between the two types of photoreceptors, the rods and cones, and why the fovea centralis is going to be able to detect better pictures compared to the rest of the retina. So first, rods are going to be more numerous at the peripheral region of the retina. They are going to be located away from the macula lutea, and they are operating in dim light. They can operate in dim light. They will provide you with a non-colored fuzzy peripheral vision. Compared to the cones, they are going to be present in the macula lutea, concentrated in the fovea centralis, and they can operate in bright light. Those will provide you with a high acuity colored vision. All right, so if we're looking on here, in order to understand, to have a better understanding of what is the difference between rods and cones, try to imagine you are sleeping in the middle of the night and you felt that somebody is standing behind the door of your room staring at you. So you wake up, turn on the light. And when you turn on the light, what appears, actually, this was not a person looking at you, it was your coat hanging behind the door of your room. So in a low light condition, if I did have a low light condition on here, am I able to see something? Am I able to see something here? Yes, I can see a person staring at me, but is it accurate? It's not accurate. But when I turn on the light, I can see what's exactly is behind my door. So who was operating in the dim light, in low light condition? It was my rods. They did provide me with a non-colored fuzzy picture compared to when I turn on the light. Who is going to be operating now? It's going to be my cones. And if the cones are operating, they will be providing me with a high acuity colored vision. So I can see the details. I can see that this is a coat that hanging behind the door of my room, not a person staring at me. All right, so remember, cones are the ones which are gonna be detecting colors. Why cones are detecting colors? Simply because we don't have a single type of cones. We have three different types of cones which are detecting three different wavelengths of light. So light travels in waves, and each color gonna be determined according to the wavelength of the light rays. So we have red cones, we have green cones, and we have blue cones. And by combining all the three of them, your brain can detect all the various colors that you can see. What if I have light that is infrared or ultraviolet? Can I see those lights, ultraviolet or infrared? No, I don't have receptors to detect them. All what's in between can be detected by my photoreceptors. All right, questions? So if you put your hand in front of your face, so put your hand just in front of your face like this. Look at your hand. How many hands do you see? If you put just one hand in front of your face, how many hands do you see? You are looking at your hand. How many pictures of your hand do you see? What do you think? So look at your hand, not something else. Look at your hand. You have your hand in front of your face. How many hands do you see? While you're looking at your hand, not at the screen. 1. Anybody else? Anybody else is trying this? One. So I can see one picture. Now look at the screen of your device while you're still having your hand in front of your face. Now you're looking at the screen. Make sure you have your hand between your face and the screen. How many hands, how many pictures of your hand do you see? I know that your hand gonna appear fuzzy, like it's not clear. But how many pictures of your hand do you see? You see 2 pictures of your hand. How come you see 2 pictures of one hand that you have in your front of your face? How many pictures do you detect for any object that you're going to be looking at? How many cameras do you have? We have 2. We have 2 eyes. So how many pictures am I detecting of any objects that I will look at? How many again? So 2. I'm detecting 2 pictures from everything I'm looking at. Why I don't actually see 2 pictures? Because yes, you're detecting 2 pictures, but your brain gonna be overlapping those 2 pictures into one. So if I'm looking at a far object, the 2 pictures that will be arriving to my eye gonna be very close to one another, so it's easy for my brain to overlap. What if I'm looking at a close object? If I have a close object in here, and I'm looking at a far object, and I have this close object closer to me, so the 2 pictures that will be detected by my eyes are not close enough for my brain to overlap as one picture. So my brain will do what? Will see 2 pictures of the same object because it did actually receive 2 separate pictures. And it failed to overlap them. So in order to prevent this, when you are looking at a close object, what you're gonna do, you're gonna be rotating your eyes medially. We call this medial rotation of the eye is convergence. You converge your eyes for you to detect 2 pictures that are close enough for the brain to overlap into one. And this is the idea of 3D movies. So when you go to a theater, You are attending a 3D movie. Do you remember what was the first 3D movies that you've ever seen? You went to this theater to watch. You remember? Doctor Strange, and for the rest of you, Disney. So for me, it was Spy Kids. This is the very first 3D movie I've ever seen in theater. Before Avatar, how the 3D glasses did look like. You know, the Avatar movie. So the glasses, the 3D glasses, starting from Avatar, maybe, it was great till now. But before that, the 3D movies did have glasses that will come with it. And one lens of the glass was red and the other one was blue. Right? Did you see those ones before? When you take off the glasses, when you take off those glasses, what you will see on the screen, you will see the main picture. So whatever here, let's say a dog here. And you will see the same picture in red and another one in blue. Like this. So what is the idea here? So the idea is that I'm giving two different pictures to my eyes. So the blue here is going to be reflecting the blue picture, so you will not be perceiving this one. You will be receiving the main picture, the colored one, and the red one. And the other eye will be receiving the main picture and the blue one. So I've received two pictures. One, which is colored from this eye, and another one which is going to be the red. It's not exact, it's very close to it. This one will allow you to see the colored picture and a slightly moved picture, which is the blue on here. What your brain will do, your brain is going to be receiving those two pictures and they are close enough so the brain will be able to overlap them, creating a depth of the picture. Although everything is on a flat screen, nothing that you're looking at has any depth for. You are just tricking your brain by giving each eye as a different picture than the other that when your brain overlaps, it creates a depth to the object, which actually doesn't exist. I'm creating a depth to the object by giving the brain two different pictures, but close enough for the brain to be able to overlap them. All right, does this make sense? All right, great. So we're looking here, this is my eye. I'm now looking at a far object. Here, let's say it's a candle. This candle is emitting light rays like this. So which light rays are going to be reaching my eye if it was a far object? The ones which are parallel rays. See here, the ones that will actually reach my eye are parallel light rays, compared to if I'm looking at a close object. More divergent light rays gonna be reaching my eye. So it's gonna be more challenging for the eye to converge those light rays together for them to fall on the retina. Compared to here, I don't need much convergence power to converge those parallel light rays. So what would I do in order for me to converge those divergent light rays that are emitted from a close object, a near object? So what will I do here is I will be laxing the suspensory ligaments holding the lens of the eye by contracting the ciliary muscles in the ciliary body. And when I lax those suspensory ligaments, the lens is going to become more convex. So if I am pulling the suspensory ligaments like this, the lens is going to be stretched and it's going to be less convex. In a closed vision, I will be laxing those suspensory ligaments. And as I lax those suspensory ligaments, what's going to happen to the curvature of my lens? It's going to be increased. You increase the curvature and thus the convergence power. So now I have better convergence than before. And this is something that everybody will notice. So you'll see your grandfather, your father will be buying reading glasses. Why would they need reading glasses? Because over time, with age, this lens is going to become stiff. So it will fail to accommodate. In other words, you can't increase the lens curvature. It's going to remain like this. So in order to increase the convergence power of the lens, for me to focus those light rays on my retina, and for the vision to not be disturbed, what will I do? I will add a lens from outside to converge those light rays before they reach my own lens. So you see on here, would you need those reading glasses if you are not reading, you are driving, for example? Would you need those reading glasses? What do you think? Would I need reading glasses if I'm driving? No, why? Because you don't need extra convergence power. I don't need it in here. It's easier if you're looking at a far object to converge the light rays on your retina, even when you are old. But what is your problem when you get older is with a close object. In a close object, I will need a greater convergence power to focus those light rays on my retina. Easy, easy. What do we call this process again? It's accommodation. So seeing far objects from far away. So do you mean the person can't see or can see? Can see a far object from a small object from far away. If a normal eye can see it and a person can't see it, so he has myopia, he has short-sighted, as you see on here, I have a myopic eye. So if we're looking at this graph on here, on this diagram, light rays, they would be converged. They are gonna be falling on the retina. This is a normal eye, but this person has a longer eyeball than normal. So the retina is located behind its normal position. So this is the retina on here. It should be on here. This is a new position of the retina. So the light will not be focused on the retina, so it's going to be disturbed. So I can't see well. Do I need extra convergence power, or would I need to weaken the convergence power in this case? If I have myopia, if I have nearsighted vision, I see better the close objects. Why close objects? Because they are emitting divergent light rays. Compared to a far object, I have parallel light rays, so the convergence is taking place before the light reaches the retina. So it's not converged on the retina. For you to see an object clearly, it needs to be converged on the retina. But here in this case, it's not going to be converged on the retina, it's going to be converged before the retina, before it reaches the retina. So what do you need? Do you need extra conversions or do you need less conversions? You need less conversions. So I will be using a lens that will be diverting the light rays. So rather than receiving those parallel light rays, I will be diverting them before they reach my eye because I have more conversions than what I need. Do you have an actual problem with the lens of the eye? You don't have an actual problem with the lens of the eye. What you do have is a longer eyeball. So the eyeball itself is longer than it should be. So the light gets converged before it reaches the retina. So the picture of a far object is not going to be clear because the light is not focused on the retina. All right. So a person with myopia, nearsighted vision, he will see better if he is looking at a close object. Why would he see better if he is reading? You will see a person, he is getting very close because he doesn't have his glasses. He is getting very close from the book for him to be able to see better. Why? Because the divergent light rays gonna be better for him because they will be falling on his retina. His retina is located behind the normal side. The exact opposite will be for the hyperopic eye. A hyperopic eye, the eyeball is too short. So the light is not going to be focused on the retina, it's going to be focused behind the retina. So the retina is, my retina is located in front of the normal location of the retina. So would I see as good as normal if the light is not focused on the retina? No, I can't see it. I can see well because the light is not focused on the retina. So do I need extra conversions or less conversions than normal? The light gets converged after the retina. So I need extra convergence power. I need to add a lens that converges those light rays before they get into my eye. So would this person see better a close object or a far object? What do you think? Looking here, this person has a problem with what? With converging the light rays. Which one is going to be emitting more converged light rays? A far object that emits parallel rays or a close object that emits divergent light rays? Which one is better for this patient with hyperopic eye? His problem is in converging the light rays. Which one is going to be easier to converge? The parallel rays or the divergent rays? So it's going to be easier for him to converge the parallel light rays. That's why the other name for the disease is going to be far-sighted vision. Because it's easier for him. He sees better a far object. Make sense? Make sense? But we can have a break for 5 before we discuss the hearing. Right, we can have a break for 5 just to refresh, and then we're gonna be back to discuss hearing and equilibrium. Right? Any questions before we go to a short break? Any questions? All right, we'll see you in 5. All right, welcome back. So we're starting with the hearing. In order to understand hearing, we need to understand two main properties of the sound. We need to differentiate between the pitch of a sound and the loudness of the sound. Pitch of the sound is going to be determined according to the frequency of the sound waves that will be created, compared to the loudness of the sound is going to be determined according to the amplitude of the sound wave. So for example, if you have a guitar, are all the chords of the same thickness? What do you think? Are all of the same thickness? No. Some of them are thicker than others, right? So as I vibrate those thin chords, they will have faster oscillation. So they are going to be able to vibrate faster compared to the thick ones. They are much slower. And that's why you have the different pitch of the sound emitted from those different chords. Also, you see on here, the strength by which you're going to be vibrating the chord is going to be determining how loud is the sound that you're going to be producing. So the more force that you're going to be applying, the louder the sound will be. So again, what's going to be determining the pitch of the sound? It's going to be the frequency of the sound waves, compared to the loudness is going to be determined according to the amplitude of the sound wave. In order to understand hearing, we also need to have an idea about how the anatomical structure of the ear. The ear is going to be divided into three main parts. We have the external ear, which is formed by the ear pinna that will be collecting the sound waves to be conducted into a canal. We call this is my external auditory meatus or tube. Those sound waves that will be collected will be reaching the tympanic membrane. Or the eardrum. The vibration of the eardrum gonna be conducted to the middle ear, where you're gonna have three tiny bones, three ossicles. We call those ossicles, those are my malleus, incus, and finally stapes. So we're looking here, external ear is going to be formed by the ear pinna or auricle, external auditory meatus or external canal, and the sound waves that will be collected from the external auditory meatus will be allowing the vibration of the tympanic membrane or the eardrum. The vibration of the eardrum is going to be transmitted to three tiny bones in the middle ear. Those are my malleus, incus, and stapes. If you notice on here, the middle ear is going to be connected to the pharynx by the pharyngotympanic tube. And this allows you to equalize the pressure on both sides of the tympanic membrane to allow free movement of the eardrum. The vibration will then travel from the middle ear to reach your internal ear. Your internal ear is formed of three main organs. First one is going to be the cochlea. Second one is the vestibule. And the third one is going to be my semicircular canals. Each of the three is going to be responsible to detect a different sensation than the other. The organs that's going to be responsible to detect hearing is going to be the cochlea. This is where I have the organ of Corti, which is the receptor responsible to detect hearing. So the vibrations have traveled from the stapes, and now they are moving the fluid inside the cochlea. This is a transverse section of the cochlea. If you notice, the cochlea is going to be divided into three main compartments. Those are the scala vestibuli, scala tympani, and in between we've got the scala media. And inside the scala media, this is where I will have the receptor responsible to detect hearing. In order for me to hear a sound, what's going to happen? The vibrations transmitted from the eardrum to the malleus, to the incus, to the stapes, and then will travel through the oval window to reach the fluid of the scala vestibuli. And in order for me to hear the sound, it needs to travel from the scala vestibuli to the scala tympani, passing through the scala media. Why do I need to pass through the scala media? In order for me to reach the organ of Corti. And the organ of Corti on here that I see, it's going to be the receptor for hearing. What is the structure of the organ of Corti? It's formed of hair cells, as you see on here. Each of them has cilia. And attaching the cilia to one another, you've got a gelatinous membrane. We call this gelatinous membrane is my tectorial membrane. So what happens? The sound waves will be traveling through the scala vestibuli, passing through the scala media, and as they pass through the scala media, they will allow the vibration of the tectorial membrane. I will be vibrating this gelatinous membrane that is attaching the cilia of the hair cells. This vibration of the tectorial membrane will cause the cilia of the hair cells to vibrate. And the vibration of the cilia of the hair cells is what's going to allow the opening of the sodium channels that will cause the depolarization of those hair cells. So those hair cells will depolarize, and as they depolarize, the electrical signals gonna be summated and collected by the nerve fibers to be conducted towards your brain for you to be able to hear. So again, how do you hear? First, the ear pinna will be collecting the sound waves that will then be conducted to the external auditory meatus, causing the vibration of the eardrum. Vibration of the eardrum can allow the vibration of three tiny bones located in the middle ear by malleus, incus, and stapes. Remember, you need to equalize the pressure on both sides of the tympanic membrane. That's why your middle ear is going to be connected to your pharynx through the pharyngotympanic tube. This vibration is going to be conducted to the cochlea located in my internal ear. When I have a cut section of the cochlea like this is how it's going to look like. You have a membranous tube. Outside of it, you've got the fluid that we call exolymph. Inside of it, you've got a fluid called the endolymph. This will allow you to divide the cochlea into three main compartments. We call this one is my scala vestibuli. This one is my scala tympani or tympani. And the middle one here, this is called my scala media. In order for you to hear a sound again, you need to travel from the scala vestibuli to the scala tympani, passing through the scala media. What if it's too soft? The sound is too soft, or it has a very high frequency. It will fail to penetrate the scala media. And if it fails to penetrate the scala media, what's going to happen? You won't hear the sound. Why? Because you're only going to be detecting those vibrations that will reach the scala media. Why specifically the scala media? Because this is where I have the receptor responsible to detect sounds. What do we call this receptor? It's my organ of Corti. The organ of Corti is formed of hair cells, and attached to the cilia of the hair cells, we've got a gelatinous membrane. This is called my tectorial membrane. Again, those vibrations will cause the movement of the tectorial membrane and thus the movement of the cilia of the hair cells. When you move the cilia of the hair cells, what happens? You're gonna be depolarizing those hair cells, and those electrical impulses gonna be collected from the hair cells and will be conducted towards your brain through the cochlear division of the vestibulocochlear nerve. All right, so what do you mean by vibration? What do you mean by vibration here? Five, 7, 2, 4, 7, 3. To the hair cells on here. So the vibration of the cilia here, the vibration of the cilia, the kinking of the cilia is what causes the depolarization of those hair cells, and those electrical impulses that you're going to be forming in those hair cells going to be conducted through your fibers to reach the brain for you to be able to hear the sound. All right, so I will be posting a PowerPoint presentation for you on Canvas that has all the most important diagrams that you should be preparing for this upcoming lab exam that will be available starting on this Tuesday. All right. Any questions for me today? Any questions? All right. Thank you so much for attending the meeting today, and we'll see you on Thursday. So again, I will be posting a collection of the most important diagrams that you should be preparing for this upcoming lab exam as a PowerPoint presentation for you on. Is there any practice exam for this lab exam? Yeah, this is the ones that I will be posting for you. It's not going to be an exam, a practice exam. It's going to be just the collected diagrams from both the central nervous system and the special senses. No, only central nervous system and special senses. Only central nervous system and special senses. Only diagrams. Those are the ones that will be covered in this lab exam. For the lecture exams, as it mentioned, like for the lecture exams, we still have. So we're done with chapter 11. We are done with chapter 12. You have few homeostatic imbalances that you're going to be reading on your own, and if you had problem with them, just ask me. We did skip chapters 13 and 14. We are now discussing chapter 15. Next class, I will be completing chapter 15 and 16 and 13 for you, and chapter 14. Next class, I will be completing chapters 14. We've already completed most of 15 and 16. So chapter 14 is just 6 or 7 slides, so it's not a big chapter. So next class, we're going to be completing 15, 16, and 14, which are included in lecture exam 4. For the lab exam, it's not going to be proctored anymore. For the lab exam, it's not going to be proctored anymore. So I did have issues with the proctoring exam with my AP2 class, so I don't want to have the same issues with you. Lecture exam 4, it's gonna be covering chapters 14, 15, and 16. We are done with most of chapter 15 today, special senses. Next class, I will complete chapter 15. I will complete chapter 16 and 14. Those are the chapters covered in lecture exam 4. For chapter 13, this is going to be completed Thursday. This is going to be our last meeting. So we're meeting Tuesday, we're meeting Thursday. On Tuesday, I will be completing chapters 14, 15, and 16. On Thursday, next week, we're going to be completing chapter 13. Chapter 13, you're going to have only the quiz for chapter 13. You don't have it as part of your lecture exams. For Emmanuel, you're asking about the extra credit. Extra credit, so we've got 5 extra credit points. I will tell you about next week, about this next week. Those gonna be for completing uh, the teaching evaluation on my FSCJ, and you're gonna be able to submit this with your in-class activity, in-class activities file. So it's gonna be embedded in your in-class activities file. Also, in-class activities, I have seen a lot of people who have trouble, I'm not sure why, to attend the recorded and the live. So the in-class activities, if you are completing them, all of them, they will be curved. So for those people who didn't complete any, so I will look at this is something that it's not ready yet, and I can't tell you exactly how it's gonna look like. I need to collect the data first from you. I need to collect all the in-class activities from everybody, and I will be comparing all of you, all of the work provided by each of you, and I will be taking the average, and this is gonna be the minimum on here. So if you are above the average, all the extra point, all those gonna be extra points that will be added to your grade. So for example, we have, and I'm not sure of the numbers, we've completed, let's say, 13 in-class activities. And I've seen that the average of the in-class activities has been 7, because a lot of people are not attending. A lot of people are missing the live meeting, are not willing to spend the time to go over the recorded lectures. So what will I do? I would be considering those 7 as the average for a full grade for 100 points. So if a person did complete 7, he will be getting the 100 points. And if you did complete the 13, each one of them gonna be for 5 extra credit points. All right, I'm trying to be fair, and in the same time, I'm not willing to be so tough in my requirements. All right, so this is what I will be doing just to accommodate those who are not able to attend or uh, or watch the recorded sessions. All right, does this make sense? Good. Questions? Questions? All right, thank you so much, and have a good night, all. Sorry for taking more time than scheduled, and we'll see you all on Tuesday.